Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at Xtalks.com. And this week, I'm delighted to introduce a very special guest on the show. We have with us today Brian Atwood, who is the Chairman and CEO of CERo Therapeutics, which is a company pioneering a new generation of autologous T-cell-based therapies for both hematologic and solid tumor cancers. Prior to joining CERo, Brian Atwood served as chairman of the board of directors of Phoenix Biotech Acquisition Corp, which merged with CERo Therapeutics to form CERo Therapeutics Holdings, Incorporated. Immediately prior to this in 2015, Brian co-founded Cell Design Labs, a biotech company focused on developing human engineering technology for the treatment of multiple diseases, including cancer, where he served as president and chief executive officer until 2018, when it was acquired by Gilead Sciences. Brian has served as chairman on the board of directors of numerous other biotech companies, including Immune Design, Veracyte, 5 Prime Therapeutics, and Biomedicines, among others. Brian holds a BS in Biological Sciences from the University of California, Irvine, a MS in Ecology from the University of California, Davis, and an MBA from Harvard Business School. To learn more about the current landscape of T-cell therapies and the exciting work that Brian is leading at CERo, let's turn to Brian Atwood now. Thank you so much for being on the show.
Thank you.
So, Brian, I'd like to start off by asking you more about the mission and goals at CERo Therapeutics.
So we are an anti-cancer company. We're developing a new generation of engineered cell therapies for the treatment of cancer. And at this point in the company's history, we are right on the cusp of applying to the FDA for the right and the ability to start treating patients in a phase 1 clinical trial. And our intention is to be treating patients with our first therapy. It's called CERo 1236 by the end of this year, 2024. And so the whole basically focused mission of the company at this point is to take our most advanced clinical candidate and putting it into the clinic in the treatment of AML patients.
And I'm going to get back to you about the indications that you're pursuing your candidates in, including AML, which is of great interest to me, and we'll discuss that further. But I'd like to actually first ask you also more about CERo in terms of what attracted you and what led you to join this particular company.
So my experience in cell therapy really began with that prior company that we talked about, which is a Cell Design Labs, a company I started working on and was a co-founder of back in 2015. It was my first really thorough introduction into engineered cell therapies with a noted academic researcher who had done a lot of work on the engineering of cell therapies. And so all of my learning came from that particular experience. It was a really good experience from founding. Actually, my first interaction was March of 2015. We were more or less founded and financed the day before Christmas of 2015. And from there, built the company to about 60 people and wound up selling it to Kite and Gilead. Just really 2 years later, less than 2 years later, it was the middle of December of 2017 that we actually sold it. We made a lot of progress in that time scientifically. I learned a lot about engineered cell therapies. I'm very enthusiastic about this new wave of therapeutic technology in trying to kill cancers and kill tumors. And so when I was introduced to this by a mutual friend, that is to CERo Therapeutics, back in about the spring of 2019, I liked the idea right away. I saw it as a next generation compared to what I was doing at Cell Design Labs. And also what I saw other companies doing. There are a lot of engineered cell therapy companies. They're in a category called CAR T. And this was a big step beyond CAR T. One of my colleagues that I work with here calls it CAR T plus plus. I like to think of it as being completely different from CAR T in the sense that it's harnessing more of the immune system to be a more powerful killer of tumor cells. And that's certainly what we've seen in preclinical experiments in the accepted models, in the accepted animal models of cancer, which are in mice, and also theoretically with respect to the actual design of the engineered cell therapy. So I really like this science a lot. And I'm really passionate about seeing it into the clinic and seeing if it works in human cancer patients.
And can you tell us more about the specific technology in terms of what distinguishes it from others in the field?
Sure. So I think one of the ways I like to describe this technology to people who are not scientists or who are not biologists is to talk about where we are historically in the treatment of cancer. And I like to think of this as the age of immunology in cancer. For about 30 years now, there have been several different important initiatives in the harnessing and the using of the immune system and the manipulation of the immune system, human immune system, in order to go after cancer. This has led to products that are generating probably north of $40 billion a year right now that have just come into being in the 21st century, really. And so this is what we're doing is part of that. To understand specifically what we're doing, we like to talk about the very genesis of the company's technology. It came with our founder, Daniel Corey, who's a hematologist oncologist, and he's also a scientist. And while he was doing his work as a hematologist oncologist, he started thinking about a part of the immune system that we probably all know about from high school biology. At least that's where I learned about this in high school biology. It goes to a part of the immune system that cleans up old and dying cells that have been infected by viruses, that actually destroys and gets rid of bacteria, parasites. So all of this stuff that happens in our bodies that winds up with biological material that's dead or dying or needs to be cleaned up, basically taking out the trash, is an important part of the immune system. It's actually called one of the two arms of the immune system, the innate arm of the immune system. This is where a cell type called macrophages exist. And macrophages, along with some other cells, like something that's called a dendritic cell, these all come from monocytes. These cells all have as their reason for living the phagocytosis or eating or engulfment of stuff that needs to be gotten out of the human body. So example, again, dead and dying cells that have been infected with viruses. And so our founder started thinking about, what if I could figure out how to basically employ the business end of a macrophage. That is, the macrophage has to actually touch or bind to a target that it wants to eat and get rid of, because that particular cell needs to be gotten out of the body. And so take that receptor and then clone it into basically the workhorse of cell therapy, which is the T cell. So it's kind of like peanut butter and chocolate. That's one of the analogies I like to use. We're combining two different parts of the immune system and creating a more powerful cell therapy-based killer of cancers. And that's what this is all about. It's the first time that anybody's actually combined both the innate and what's called the adaptive sides of the immune system, and has created something. Another metaphor that I like to use is kind of a Swiss Army knife. This particular cell therapy that Dan Corey invented along with his team has about 8 to 10 different functions, including the way that it kills a particular target cell. It includes eating the target cell. It actually includes punching holes in the target cell. And it also has other functions in terms of communicating with the rest of the immune system, both distant and as well as nearby, to engage the rest of the immune system to go after the cancer and actually teach the rest of the immune system what the cancer is made up of. So there's a lot of complexity here. One of the things that we've had a hard time doing, frankly, in communicating what our cells, we call them CER T cells, CER, T, are all about is that they do so many different things. It can be kind of bewildering. But the best way to think about it is that our cells combine the most important parts of the immune system to actually go after a tumor cell, to target a tumor cell and kill it, and then talk to the rest of the immune system and say, hey, there's a cancer here, bring everything you've got and help us kill the tumor that we're going after right now. That's why I'm excited about it. It's the first time it's ever been done.
That is indeed very exciting because it is a very novel approach compared to the other T cell therapies that you hear about, and even the next generation ones where they're adding more and more components of the immune system, but to have components of the macrophage, which intuitively makes sense. And for CERo to be doing it, that's very exciting. And I'd like to know what indications your lead candidates are being pursued. And I know that you mentioned AML, which is one of the most difficult cancers. I've worked in this space for my graduate studies. And so I would love to hear more about that.
Sure. So our first indication is AML, or acute myeloid leukemia. This is one of the most deadly cancers that exists. I think if you don't know cancer very well, people who are worried about it or scared about it would think of, say, brain cancer or pancreatic cancer. For sure, AML ranks up there with those. If you wind up with this disease, it's almost certainly a death sentence. It is a disease of the blood system, and it is also a disease of older people. And so as a result, the therapeutic options available to the vast majority of patients is fairly limited, very limited. So the reason why we're going after this is it's a place where, first of all, our particular technology has very unique applicability, but also if we're able to demonstrate at least a little bit of efficacy, activity in this particular cancer, then it probably opens up for us a path through clinical trial work and towards FDA approval that's a little bit better than, say, in other cancers where there's so many other therapeutic options available, for example, in breast cancer. And so this is a particularly important cancer for us. Somebody would call it a low-hanging fruit. I don't. AML's hard. It's complicated. The patients are moving pretty quickly in their disease, so you have to catch them fairly early. But we've studied the disease very carefully and feel really optimistic about being able to get to a signal in this patient population if there is a signal to be had. But I guess the other thing I'd mention here is that we have a target for our engineered T cells. And this target is found on AML tumor cells. And that's really important. It's expressed at a high enough level compared to normal cells that we actually target the AML cancer cells very effectively. We've seen that in our preclinical studies in mice. The same target is found on other cancers. And so we think we have an opportunity inside baseball. It's called pan cancer, but we think we have an opportunity to go after lots of cancers. This particular marker or target that's found on these cancers is found on ovarian cancer cells. It's found on the cells of non-small cell lung cancers, and also on in the area of B-cell malignancies, for example, mantle cell lymphoma. And if you look at the literature, this particular target is found on other cancers. So we think this particular target is overexpressed across a wide variety of cancers. And so we'll be starting with AML, but we'll be branching out relatively quickly to the B-cell malignancies, where we know we have the overexpression of the target. And then after that, we're actually going to be working on an IND for ovarian cancer. And we'd hope to have that either well underway by the end of 2024 and filed in 2025, or hopefully even in 2024. So we're going to move along pretty quickly here to branch out from AML.
Wonderful. And are you able to share what that target is, or?
Yes, it's in the scientific papers that have been published. We have two major scientific papers that have been reviewed and published in the past year, and also it's in our investor deck which is provided in on the website. Also, the SEC filings have this in extraordinary amount of detail. But it's called the TIM4 ligand. And more specifically, it's a ligand that's called inside the scientific discipline the eat me signal. So it's the actual target that's expressed by dead and dying cells. It's one of the ways that the immune system knows, that's a cell that I want to get rid of. So think back to COVID, and you've got an infected cell. One of the things that it pops up is this particular target, the TIM4 ligand, the eat me signal, and that way the immune system knows, okay, this is a cell I need to get rid of.
Great, thank you. And so I think just maybe taking a step back, what are some of the current challenges and barriers to entry in developing and commercializing T-cell therapies? I know the field has come such a long way in just, I think, the last decade alone. But with that evolution, are there new challenges? Are there existing challenges, unmet needs, and all of those things that can come into play? So what are those in your opinion at the moment?
So by far the most discussed would be manufacturing. These are this is a complicated manufacturing process. It was wildly more complicated back in the early 2010s when these therapies were first being used in human patients. And so we've actually come a very long way. But the manufacturing technology here begins with the withdrawal, if you will, of a patient's blood, and then the T cells from that blood. Those T cells are then transformed using genetic engineering techniques in order to be focused cancer killers. They're grown up so that you have lots of those cells available, basically something in the neighborhood of 100 million or so, or even into as many as 1 billion, put back in a bag and then transported back to a patient. That whole process can take almost a month. Used to take longer than that back at the very beginnings of this particular wave of technology. But these days, what they call vein to vein time is something in the neighborhood of 3 weeks. So from the time that you withdraw the blood to the time you reinfuse the patient with the transformed T cells takes about 3 weeks. The actual manufacturing process is about a week. There are so many people working on this that I think by the time we get to commercialization with our product, if we're lucky enough to get there, we'll have the vein to vein time probably well less than 3 weeks, maybe closer to a week. But cancer patients are anxious, their practitioners are anxious, we're anxious. And so I think we're all interested in getting that manufacturing process better and better. We have no problems right now at the numbers of patients that we're going to be treating, which is a couple of dozen in phase 1. We have adequate support from a contract manufacturing organization, and so we'll be able to do that. But we're going to be watching very carefully the development of technology from vendors in this particular area to make sure that this vein-to-vein time, this manufacturing cycle comes down, and with it, hopefully, the cost. I think the other kind of classic problem with this technology has been trying to find a target that works for it with respect to cancer. The first target in the B-cell malignancies was CD19, and that worked really well for that class of cells. But other approaches to pick targets in other liquid tumors or blood system tumors like AML, and also in solid tumors, haven't been as fortunate just because of the kind of the heterogeneous nature of markers in cancer cells. We think we've got that beat. The eat me signal, as I like to think, is kind of an Achilles heel for cancer cells. Our founder likes to talk about the fact that the TIM4 ligand is a stress signal. It's a signal that a cell throws up to indicate to the outside environment that it's under stress and it needs to be cleared out. Cancer cells live in stress. They're just very, very dynamic cells. And that's why we think, we don't know for sure, but that's why we think that they're throwing up the TIM4 ligand signal. And therefore, we're taking advantage of something that cancer cells are doing that let us target them specifically as opposed to normal healthy cells and go after a tumor more effectively. Again, that's the theory. That's the mechanism of action. And that's what we've actually seen in our 10 or 11 mouse studies across several different cancers.
And so now you're preparing for your lead candidate to enter phase 1 trials, as I
Right.
understand? Okay.
Yep.
And when will that be happening?
But by the end of the year,
By the
we
end
have
of
to
the
file the IND first. The IND is moving along very quickly now. That's what
Right.
everybody in the company is working on. We hope to have that filed in the near term. And then, as you probably know, that once filed, that goes before the Food and Drug Administration. They have a 30-day review and approval window, and we're hoping we'll get through that successfully. If we do, then that means after 30 days after submission, we can start up the process of getting our clinical trial sites engaged and rolling.
Yeah. Wonderful. And I'd like to ask you more of probably a broader or more of a question that taps into your personal expertise, experience, and again, your prolific career in the biotech space, having worked in so many different companies, leading, and so I just wanted to ask in terms of what are some of in the rapidly evolving field of biotech, how have you seen the space evolve in your time in the field? And also, when you're working at a particular company, how do you really foster that innovation and how do you lead that? Both, taking into account collaboration within your company, outside of the company. And I guess my question is, what does it take to be a biotech leader, such as yourself, who has such an illustrious career, and what kind of advice would you give people aspiring to be someone like yourself?
So one of the things I like to talk about in terms of being a member of the broader class of startup companies is startup companies are all about focus. The reason why they're successful is we're going to do one thing. And we're just going to drive like hell to get to that one thing. And so that's what I think marks a great company is the leader understands that one thing. Where's my IND? And then talks to the team and says, look, folks, we're all in this together. We got to drive to this one thing. I think companies lose their way when they think about all the wonderful things that the science can do. And I'm kind of guilty of this as well. I love the science in this business. It's the reason I'm in it. I was a biologist all my life, all the way back to when I was a kid. And so it's easy to be seduced by all the different things that your particular corner of science can do. But you have to kind of keep away from that. You have to steer away from that and have the culture of the organization oriented towards, we're going to focus the science on one mission at a time. So currently here, it's where's my IND? And then once we're filed here in the next few weeks or a couple months, then where's my first human patient result? And where's my second human patient result? We'll do other things, but they'll be secondary. And we'll do those things as a part of the culture in order to enhance what we're doing as a team of scientists. I'm not, but that's what I have here is a great team of scientists. But what I really think about is, and everybody here thinks about it, it's like, this is a really cool invention. We just want to see if it works in a human cancer patient. And everybody's really dedicated to that. That's kind of the curiosity. That's the passion here. And that's what I think I strive for as a leader in these companies. It's kind of like, what's that next step? And let's get everybody on board to going after that step.
That's wonderful insight and advice. And so also in terms of, like, I think you mentioned, the focus that needs to be there. And so how do you balance having, let's say, short-term milestones with long-term strategic goals? Because the long-term, I guess your short-term milestone at the moment is where's my IND? And then so the long-term, how do you, it goes with the flow, I guess, but how do you balance sort of the short with the long-term?
To be honest, here it's pretty easy because we've talked about this before for the CERo story. Because the marker that we're going after appears in lots of different cancers, the way I was trained in business school is in terms of frameworks that are really important for managers, and one of them is decision trees. And so I tend to look at what sits in front of this company with its clinical lead candidate, CERo 1236, as basically an expanding decision tree. So if we see this particular result in this type of AML patient, then we walk down a different branch, say if compared to another particular result. And the same thing's true about kind of borrowing this from another industry, the product roadmap, because we have all of these preclinical studies that show the marker is on B-cell malignancy tumors, and it's on ovarian cancers, and it's on non-small cell lung cancer, then I know what my product roadmap looks like. And so the next place I'm going to be filing an IND is ovarian cancer, and then after that, non-small cell lung cancer. And the team will find out within those particular big cancer categories which specific subtypes of ovarian cancer patients we want to go after to see if we can get a signal in a place where there's a market. So I tend to think about what you're talking about as being a decision tree. And the decision tree unfolds in front of you as you get results in the clinic. And the art of the decision tree is to start with the cancer where you're most likely to get a signal, so that it opens other doors or opens other branches in the decision tree. That's kind of the way I'd answer your question.
Yeah, that makes total sense. Thank you. And so I guess just to round off, I know that you talked about some of the things that we can expect from CERo in the coming months. Hopefully, fingers crossed for that IND for sure. And so what are some of the longer term goals? What can we expect in by the end of the year and into next year?
So we're hoping to be in something between 3 and 5 patients by the end of this year. I really don't have better numbers than that, but 3 to 5 patients would begin to demonstrate to us some of the biology of the clinical candidate in our AML patients. And then we'd hope to be able to get through the remainder of the 25 patients by the end of 2025. My clinical development colleagues tell me I'm nuts, but I'm going to be pretty pushy for that. And I think also our principal investigator at our lead clinical trial site, can't say who it is right now because we don't have them under contract yet, but this principal investigator is very enthusiastic about taking this into AML patients. The hospital system where he treats patients he says 400 patients a year that fit our entry criteria within AML. So we're not talking about all AML patients. We're talking about a subset of AML patients that are further along in their disease and have specific molecular characteristics. So having parsed down those AML patients at this particular hospital system, we're looking at one clinical trial site that has hundreds of patients available to us. So once we get a little bit of traction, and we're hopeful we will, we should be able to move along pretty quickly in recruiting this phase 1 trial.
Well, that's really, really exciting to hear. And I personally am looking forward to all that's going to be coming out from CERo in the coming months and the next few years. And again, I'm also very excited about CERo pursuing this in AML, which again, as you talked about, it's such a devastating disease. So thank you so much for the work you're doing, and wishing you the best of luck. And I don't think you need it, because I think you have the right focus, and it's wonderful to hear your insights in that regard. So thank you so much, Brian, for being on the show.
Okay. Thank you, guys.
All right. That's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we'll see you next time. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.